Okay, so today is the most mathematical day. So what we will see is all the possible models that we can fit to the kind of data that we will use during systems biology. And we will present a few examples of them. And the first set of models are called static network models. And a network is something like this. And already using this slide to introduce already some concepts about networks. So you may have a network is again some agents that are related to each other. And these agents can be proteins, they can be genes, they can be individuals, they can be whatever. And then the type of relationships can be of different kinds. They can be random. So any individual can be, any agent or any node can relate to any other node. Or they can be something that is called scale-free. So scale-free, we will see in more details what is. There are some local neighborhoods, and then some guy in the neighborhood is related to some other guy that is very distant in the neighborhood. And we may have hierarchical networks that are, for instance, many telecommunication networks, they are hierarchical. So there is a hierarchy of nodes, and each one of them is of a higher level. And then the nodes of level 1 can only connect to nodes of level n+1. So there are many different kind of networks. Here you have an example of network in interactomics. Interactomics, you have their proteins, and these proteins interact with each other. And here you have another example. This is a protein-protein interaction network. So this protein-protein, you have different proteins here. This is one of the proteins that we are working in the lab, ERBB2. So you see which are the connections, which are the relationships between these ones, and you can open this screen. Sorry. Okay, so you can look, for instance, you can look for one protein here and say ERBB2. And then you have protein in different species. So here in Homo sapiens, Mus musculus is the mouse. So we can see this one here, for instance. But you go to the protein. Go. Okay. And then here you have, this is your protein. And then there is a whole here, whether there are, so these are the query proteins and the first shell of interactions. So this is your query, and it directly relates to all these. And then whether they have a 3D structure or not. So all these proteins, they have a 3D structure that is known. And then you can analyze what is the relationship. And then, so there is a lot of information here. And they are in the gene neighborhood. And how do you have it, obtain that link and this kind of stuff. So how you have networks between proteins, how do you get that information? Okay, so this is protein-protein interaction networks. And the string database is one of the best known databases for this. Okay, so let's see the mathematical models of that. So we have a graph, and a graph is defined mostly by the adjacency matrix. And the adjacency matrix is a matrix that has as many rows and columns as nodes. So here we have the nodes B1, B2, B3, B4. And then you can encode here the relationships. So here, for instance, node 1 is related to node 4. So node 1 is related to node 4. And then node 2 is related to node 1 and 4. So node 2 is related to node 1 and 4. And 1 is related to 4, but 4 is not related to 1. So 4, 1 is 0. Okay, so 4, 1 is 0. Sorry. While 1, 4 is 1. Okay, so this is a directed graph. And you may have the undirected graph. So the undirected graph, the edges do not have any direction. So here 1 is related to 4 and 4 is related to 1. So here you have the 1, 4, but also the 4, 1. And so these adjacency matrices, they are symmetric. So A transpose and A are the same. And then you may have knowledge graph. So knowledge graph here, we say that the two nodes are related, but we are not saying what kind of relationship they have. So maybe V1 is a teacher and V2 and V4 are students, and then V1 is teaching V2 and V1 is teaching V4. And V2 and V4 are in the same group of students, and V2 is the group leader of that team. So V2 is the team leader of V4. So now the edges also have equality. So it is they have it is not they are related, but they are related with respect to this function. And you can also put weights, and the weights can be transferred to the adjacency matrix. Okay, and then what are properties that we can measure on graphs? So probably all these are familiar to you. So the degree is the number of nodes to which a node is related. So here V1 is of degree 2 because it is related to 2 other nodes. V5 is of degree 0, V3 is of degree 1, and so on. And you can construct paths. So a path is when you have a graph and you want to go from A to H, you can construct a path. And then this path traverses all these edges. And you may have cycles. So when you have a cycle, it is a path that is closed. So the beginning and the end of the path is the same node. So you may start in node 2, you go to 4, 3, and then you are back to 2. So there is a cycle here. And many graphs in many algorithms, they are restricted to have acyclic graphs. So graphs without any cycle. Okay, so graph properties, so we can define a connectedness. So given a graph like this, we may define which are the connected components. So these 3 nodes are connected because there are edges that you can go from one to the other. From any one in the connected component, you can go to any other node. The same here, the same here. This is a connected component of size 1. Then you can say that a graph is planar or non-planar. So planar is, a graph is planar if you can draw it in a piece of paper without crossing edges. And this graph is non-planar. So it is not only about how to draw, it is a property of whether it can be drawn or not. So that, for instance, I could turn this point here, and then the edges would be crossing. But it is simply because I draw it like that. Okay, the edges would not be crossing, but you can move it here, for instance. You can move this point here, and then the edges are crossing, but it is not because the graph is not planar. It is because you draw it in a way that the edges were crossing. But there are graphs that are non-planar by nature. There is no way to draw them such that the edges do not cross into the. And you can also have bipartite graphs. So bipartite graphs are graphs in which the edges connect nodes of different kinds. So let's say that you have 2 sets of nodes. So you have the red nodes and the blue nodes. And the edges are always connecting a red node with a blue node. So that is a bipartite graph. And why graphs are important in systems biology? Because they're, you may remember these, well, we have already seen these protein-protein interactions, but also the regulatory networks, they are also very much, they use these kind of models. So for instance, let's say that we have a gene regulation. So the X is controlling the expression of Z. So X is a gene, and the protein, the transcript of X somehow controls the expression of Z. And there, it has been noticed that there are some patterns that are somehow repeated in many biological networks. This one is one of them. So this is called the feed-forward loop. So X controls Y, Y controls Z, but also X controls Z. And this BFAN, in which X controls Z and W, and Y controls Z and W. And in neurons, there are also this kind of connection. So now the nodes are not genes, they are neurons. And who is exciting who? And you have the same kind of feed-forward loops and BFANs, B-parallel, so these kind of patterns. And food webs, and if you are interested in this topic, you can follow this paper. And this guy, Yuri Alon, is very well known in systems biology. Actually, one of the books that is recommended is by him. And you can also have different graph properties. So I can define which is the clustering feature of a node. So let's say we have different graph, and I compute this ratio. So this ratio is KB is the number of neighbors of V. So here, this node has 1, 2, 3, 4 neighbors. And this point here, or this set there, is the set of all edges. So U and W is an edge, you see, such that W is in the neighborhood of V and U is in the neighborhood of V. So somehow what I'm looking is neighbors of mine that are also connected. Okay, so I count how many of my neighbors are connected between themselves, multiply by 2, and then divide by that thing. And that is a number between 0 and 1. And it tells you how well connected a local neighborhood of a point of a node is. So in this example here, the dashed edges do not exist. Okay, so they are not, they could be, so only the solid ones are true edges. So these ones could exist, but they don't. So this node is isolated. So the neighbors of all of node 1, they are isolated. If you want to go from 2 to 5, you have to go through 1. There is no other way. So C1, the clustering of C1, according to that definition, is 0. This is the opposite. All are connected to all. And then, given any one of my neighbors, so I am neighbor of 2 and 3 and 4 and 5. So any one of my neighbors is connected to the other. So if you make this this ratio, now this ratio is 1. So it defines this clustering, how well connected is a neighborhood of a node. Okay, so you can define these things. You can also define different types of graphs. So we have already seen this random, a small world, and a scale-free. And the characteristics between the three, so random is anyone can be connected to anyone. A small world is that is a high local clustering, so locally nodes are well connected. And from time to time, there are big jumps, so there is a long connection. And scale-free is this idea that locally there are kind of clusters. So this one is a cluster, this one is a cluster, and the clusters are are the ones that can make long jumps. And that is this is scale free. They have a property that is that if you analyze what is the histogram of the degrees, so every one of these nodes has a degree. Now you calculate what is the probability density distribution of that degree. And these degrees, this probability density function is somehow an exponential, a decaying exponential. So here you have it. So here is a number of relationships. This is the degree. And for a scale free, the distribution looks like that. While for random, for instance, it is like a Gaussian. So the distribution of the degrees tell you what kind of graph you are working with. This is the same. So here you have the distributions of a random, a small world, and a scale-free networks. And for instance, scale-free networks, they are bad in the sense that they are very sensitive to failures. So if this node fails, all these subgroups become disconnected. So failure can be in a telecommunication network, can be one of the machines break, but in system biology, it can be one of the genes. You have a mutation in one of the genes, and this mutation is killing you. And yeah, so but our networks are not scale free; they're much more resilient. Okay, so here you have another database of these kind of connections. So this one, this BioGrid, let's explore how it looks like. So here we have, this is a protein, and then we can see how many other proteins. So it interacts with almost 600 proteins, and it has interacts also with 7 chemicals. And this information has been taken from these 180 papers. And here you have all the interactors. So that protein is interacting with this one, with this one. This is a small molecule. This is an antibody. And then it is interacting with this one. So this is part. This protein is involved in cancer, and then this is one of the treatments to treat cancers that have this mutation. And then the interesting part now is this network here. So this network here, the protein is at the middle. It is interacting with 600 other individuals, but with some individuals, it is interacting a lot. And then you could analyze, not by eye, this is impossible, but you can construct programs that can read these graphs and say properties of the graph and see which are important proteins. So for instance, let's see if we could have this kind of effect somehow, that locally some of the proteins are very important, and if you have failure there, then you have a problem. And yeah, Cytoscape is another database also for this kind of information. It presents the information differently, and the information content also is also different. And here, what I'm adding you is a few software that manages somehow this kind of information. So Cytoscape, we have seen it, and there are many others. So you can, if you are interested, you can follow these. There are also databases. So one thing is the software. The other one is the content. So the content is in these databases, and you have databases for signaling networks, for transcriptional networks, and protein-protein interaction networks. A little bit of everything in these unified databases. Something else that is somehow related, but not exactly. So this Phosphosite Plus is a database that tells you post-translational modifications. Then gene co-expression networks, these are genes that are co-expressed. So if one of them is expressed, the other one is also. Gene regulatory networks, genome interaction networks. So you have a lot of information there. Actually, there is too much information, I would say. It is difficult to navigate in all these databases. There are gene disease, gene drugs, again, integrated multi-layer networks. And somehow this NCBI is the National Cancer Bioinformatics Institute or something like that in the US. And it tries to gather many information from many different databases and present it in a homogeneous way. And this is a different kind of network. Okay, so this network here, they use probability. So our previous networks, there was simple connections. At most, there was a weight in the connection. Now, the connections and the network itself, each one of the nodes will have probability distributions associated. So this is I'm explaining it without any gene or protein or whatever, because I think this is very easy to understand. Actually, the resolution is not so good. So let's say this is a network that models the following situation. So you have a house, you are away from the house, but you have a couple of neighbors. You have David and Sofia, your neighbors. And the house has an alarm, and then they call you because the alarm has sounded. So now you wonder, okay, there could be two reasons for that. One is that there has been an earthquake and then the alarm sounds. The other one is that there has been some burglary, so someone entered into the house, and then that's why the alarm sounded. Okay, so we have to associate probabilities to each one of the events. So these are conditional probabilities. So let's start from the end. So the alarm sounds true or false. So it has only two states. And then this is the probability that David calls you. This is the probability that David doesn't call you. So David calls you if the alarm sounds with probability 0.91. Okay, good. And then David calls you if the alarm has not sounded, probability 0.05. And these ones are the opposite. So if the alarm sounds, either he calls you or he doesn't. Okay, so by rows, the sum has to be 1. And this is the same for Sofia. And then what about the alarm? So the alarm depends. So David only depends on one variable in the network. Okay, so that is why David, that is true or false here, only depends on one variable here, that is the alarm. But the alarm depends on two variables in our model. So we have burglary and earthquake. So it can be true, 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 false, false, true, false, false. So by rows, they have to add up to 1. So if there is a, let's say, if this is confusing. So if the burglary, if there is no burglary and there is no earthquake, the alarm will not sound with probability, so will sound with probability 0.001. Okay, now if there is no burglary but there is an earthquake, then there are sounds with probability 0.31. These are the probability of making sound, and these are the a priori probabilities. They don't depend on anyone, so these are the input to the network, and these are the a priori probabilities of having a burglary that is 002 and having an earthquake that is 001. Now we can make this kind of calculations. Okay, so what is the probability that the alarm has sounded, but there is not a burglary, not an earthquake, and David and Sophia both call us? Okay, so let's see. This is our probability that we want to compute. So Sophia called, David called, the alarm sounded, but there was no burglary, no earthquake. Okay, so we can decompose this probability, that this is a joint probability of 5 variables. We can decompose it into pieces thanks to the model. So we can decompose it. Okay, so let's see. Sophia depends on the alarm. Sophia is here. So Sophia depends on the alarm. David depends on the alarm. So these two are coming from here. So the Sophia called, this is true. And the alarm sounded, so it is this 0.75. And David called, so it is this one, so this. So David called, so it is this column, and the alarm sounded, so it is 0.91. Okay, what is the probability? So these depend on A, that is the parent of these two. So what is the probability of A? Probability of A should depend on its parent. And there is no burglary, there is no earthquake. So the probability that the alarm sounds, this should be true, so the alarm sound. When there is none of the two, is this one, 001. This is false, then this one is 0.001. And then what is the probability? So it depends on its parents, and the parents don't depend on anyone, so they are independent. And then what is the probability that there is no burglary? It is 0.998, and the probability of no earthquake 0.999. And then you compute that product and you have 0.06. So that is the probability that both of them call you, the alarm sounds, and there is no burglary and no earthquake, is really low. Okay, and you can make this kind of calculations very easily. They are also called probabilistic graphical models or causal networks. These are the names of this kind of networks. And you can use them to calculate any probability in any network that you like. For instance, here, you can have a gene that is expressed here in the nucleus. You can measure whether the gene is expressed or not. But this gene, the expression of this gene is depends on some cascade. Okay, so there is a cascade here, and the cascade may spontaneously activate. So you have to assign the probability for the spontaneous activation. And the conditional probabilities, all these before, these ones here, they're conditional probabilities. So it is the probability that David calls you, given that the alarm is activated, given the state of the alarm. So here, you have also to define those conditional probabilities. So, so what is the probability that the cascade activates if there are a couple of cellular receptors, and these cellular receptors, you have the receptor R, the receptor S, and then it may bind something from outside. So this is a signal, and this signal happens with probability 0.2. This kind of signal happens with probability 0.4, and then you define all the conditionals, and you can compute whatever probability you want. So you can say, okay, what is the probability that the gene is not activated if there are these signals, or the signals are not present, or there is a problem in the cascade, or whatever. Okay. And the kind of input for these, because you need to estimate those probabilities. So you need to estimate those tables. So you need data for that. And the kind of data that you get is of this kind. So you can measure whether in a particular experiment, what do you measure? And we measure in that experiment, this line, we measure R was not activated, this receptor was not activated, this receptor was activated, the cascade was not, the gene was not expressed, and SET, that is another kind of response, was activated. And then for another experiment, we had this response, and for another experiment, we have some other response. From those responses, you have to estimate these tables. And if you are interested in that, you can. This is a free book that is on the net, so you can look for it. And it's learning Bayesian networks, or learning in the sense of estimating the parameters. And again, as before, there are programs that can deal with these kind of models. Here you have the names of those programs. Some of them are Python libraries, if you are familiar with Python. And now we will introduce a different kind of models, and that means Boolean networks. So Boolean network is a network in which we have the protein, and we have some other proteins, and there are circuits between those. So you can represent it in this way, but and also represent it in this way. So for instance, here, you can say, okay, this protein will be activated if AKT is activated or WNT is activated, and then I negate that. So for this to activate, neither of these two have to be activated. And so you construct circuits like this, and you can also represent these relationships, this kind of networks that we saw at the beginning. But here it is telling you how they work. So it is not only that there is a relationship between these two, and telling you what kind of relationship there is. So for this to activate, I need that this one is not activated. And I can make even one step further, and I can say, okay, the activation is not deterministic as in a logical circuit, but it is probabilistic. So there is a probability. So let's say that I have 3 proteins only. None of them is activated. So I have 0, 0, 0. The probability that in the next time that I look at it, so I regularly sample it. So next time I look at it, at t+1, I stay at the state 0, 0, 0 is 0.8. And going from 0.000 to 1, 0, 0, 0 takes probability 0.2. So now I can so you may remember from here. So you may remember from the first, you had a course on electrical circuits, this kind of, digital circuits.
How do you know which network to use in which case?
You have to infer the network. You have to infer the network. That's it. That is also something we have seen it a bit here. So here we have said you have to infer the parameters, but it is not only the parameters, you have also to infer the relationships. So from this kind of data, you also need to infer what is the structure of the network. So who is the parent of whom? Or here, how do I learn which is the circuitry that is there? How do I learn that? And all you have is data. So all you have is this kind of data. So let's say that I have I don't know, several proteins. And I think that E is a function of these. So you may remember all these. And then you have some result. I don't know, 1, 0, 1, 1, whatever. And how do I infer E as a function of A, B, C, D? Do you remember there was something called backbone maps? Did you use that? On this one, we're telling you what is the function of E as a function of the others. But this is deterministic. You can make it probabilistic too. So you can make it probabilistic and say, so because the other one, the Carnot sort of this circuit, is deterministic. If there is a particular combination, the next step will be this one. But in reality, in biology, it is more probabilistic. So it is very likely that it is that one. That could be something else. So these probabilistic Boolean networks, there was a time in which they were very popular in system biology. And here you have different software that can handle this kind of stuff. So Boolean networks, probabilistic Boolean networks, and you have both problems. How to run them. So once you know the model, how to evolve it, that is the easy one. The difficult one is from the data. How do I estimate the probabilities? How do I estimate what is the structure of the network? That is the difficult. Any question up to here? Okay, so then we'll move on to a different kind of models, and these models, they are more used for these signaling networks, transcription networks, protein-protein interaction networks, this kind of stuff. So signaling networks are this kind of relationship of structures where you have a receptor in the cell surface, and then something binds on the top, and then this signal is transmitted to the inner part of the cell, and then typically it is through some kinases, and these kinases somehow they reach transcription factors that could be other intermediates in here. So there could be a long cascade of connections, and then at the end, some genes are transcribed or not. And for this kind of networks, the most common model are these metabolic networks, or are models based on differential equations. So let's say that we have these three possible reactions. So we know that these reactions occur. So if you would like to put it in a network, so you could have CP, PC, CP, and AP. And then you could say which one is related with each other. But this network, this graph, doesn't tell you much. The differential equation tells you much more. It tells you how the different concentrations evolve over time. And the models or the differential equations are modeled in this way. So the change of the concentrations over time is a matrix times the velocity of each one of the reactions. So you need the reaction velocities. So here we have three reactions, so we will have three velocities. And here CP, that the concentration of CP, we will call it X1. CP is transformed into PC. Okay, so somehow there is some rearrangement of the complex. So, and it will happen at two different rates. So the conversion of CP into PC at rate K1 to the right, and PC to CP is K1 to the left. So these are just two numbers, and the rate depends on the concentration of X1 and X2. X2 is PC. Okay, and the same for CP for the second equation. So the second equation, the velocity there is you transform C and P, that are X3 and X4, the concentration. So you transform X3 and X4 according to a constant to the right into X1, and then X1 is transformed back into C and P at a rate K2 to the left, and the same for the last. And so now you have the three velocities, and you have to multiply these velocities by how they're changing how x1 is changing over time. So x1 here, x1 has a positive. So in the first reaction, x1 is decaying. Is x1 is transformed into something else. So it will have a negative sign. And in the second one, in the second reaction, x1 is created. So according to this velocity, velocities are always written in this way, from left to right. Okay? So from left to right, Cp is created. So Cp is created, so we have a positive sign. And then in the last one, Cp is destroyed from left to right. So it will have -1. And then x2. x2 in the first one is created. And then here it doesn't appear, and here it doesn't appear. So we have 1, 0, 0. x6 here. x6 doesn't appear in the first one, doesn't appear in the second one. So we have 0, 0. And in the last one, it is created. So we have 1. We have the positive sign. And we can model metabolic networks like this. So yeah, you have seen this in biochemistry probably. So you have this is glucose is phosphorylated, and then it is transformed into fructose. And for this, you need ATP and ADP. And you can write the different velocities. So you can say velocity 1 will be, I don't know, this thing. Velocity 2 will be this one, and velocity 3. So you write the different velocities, and now you can write the differential, how they change over time. Actually, you may look at this one. Here I have separated into a matrix and velocities, but you can multiply the two, and then you have this kind of equations there. And you have to give initial, so there are some constants here in the velocities that are constant. So you have to estimate those constants. And you have to give initial concentrations. So to fix the initial concentrations, and then you can let it evolve. So you can, okay, with these initial concentrations, how do they go over time? And then you know exactly what is the profile of each one of the components that you are, of the nodes that you are studying. And here this article is very good actually. So you can also, if you're interested in this kind of stuff, you can read it. Okay, so these were closed systems. So the concentration depended only on the current concentration. But what if we have an open system? If we have an external input, and we can also excrete something, so we can take nutrients or compounds from the environment, and we can put them back into the environment, whatever metabolites that we don't want. Okay, so open systems is the same equation as before, but now you have an extra term, and this extra term is a vector, because this is a vector, so it has to be a vector of this same size, and this minus is simply for convenience. Okay, it is arbitrary design, so it is for convenience. It is more convenient like that, and you'll see why. Because something that is interesting is the steady state. So these things change over time, but they may also reach a steady state. So a state that is fixed, so it doesn't change anymore. And this is somehow related to homeostasis. Homeostasis, you are trying to keep a steady state. If you go away, you try to come back and come back to the steady state. And so if you are in a steady state, your concentrations do not change over time. If they don't change over time, then this vector is 0. So now you have that this right-hand side is equal to 0. And like that, so velocities are known. X is what you are looking for. This is the input. So these you are trying to solve for what are the levels of the different compounds at the steady state. And this technique is also called flux balance analysis. Okay. And here you have a paper. This guy is also very well known, Bernard Paulson in system biology. And here is a paper that makes a summary of how this flux balance analysis is used. So first you need this genome scale metabolic reconstructions. You need the network, who is related with whom, which are the chemical reactions. So then you have to write the chemical reactions and somehow come with this stoichiometric matrix S, the fluxes that are the velocities. And then this is a linear equation system. So you write the linear equation system and you may solve it for steady states, but you can also define objective functions. So you can say, okay, which are the conditions that make these, for instance, in a bioreactor, that make this cell culture to grow the fastest. And then your objective function is biomass, and biomass will be somehow depending on the concentrations and the velocities. So some constants and the velocities. And then you can try to optimize this thing and to see how do you have to feed that cell culture so that it grows as quick as possible. And here's some software dealing with metabolic networks. Any question? No? So I think good time for a break. We come back at 31. Okay, so let's see different kind of aspects of these mathematical models. So we have seen 3 different kind of models. So we have seen these static models. So these static models in which we have a network of relationships, but we don't have any stoichiometry. So we don't know how many. So we know that these 2 proteins relate, but we don't know if it is 2 copies of this protein with one of these. We don't know what is the stoichiometry of the reaction. There are no parameters, so we simply know that there are relationships. So this is the simplest kind of model. Then we have seen also static models like the flux balance, where we know the stoichiometry, we know the reactions, but because of the steady state, there is no change over time. And then we can include time in the modeling. And these are called dynamic models. And you will need the parameters for that. Okay, a different kind of models are also these finite state machines or automata. So very simple automata, the traffic light. So the traffic light, you have a red light that then after a time it goes to green, and then green goes to yellow, and yellow goes back to red. And each one of these colors is a state. So the system is in a particular state, and then it goes from one state to the next. And you can also do it with, for instance, a document processing procedure. So you can start with a draft, and then you are in the state draft, and then after time, next time I will be in the same state. So I'm updating the document, and then at some point I may ask for somebody else to review the document, and then there is a change of a state from draft to review, and then this person may tell me, okay, you need to change something. So there is a change of a state, and then there is another change of a state that I have to modify the graph and so on. So this kind of state models are also very useful in many different kind of processes. And we can also define finite state machines for system biology. So you have a network you have a set of genes and proteins and this stuff. And the state is defined by the concentration of each one of the proteins you are looking at, or the gene expression levels. And then you may the change how one state changes to the next is controlled by these kind of regulatory networks. And regulatory networks we have already seen. So there is a repression here, there is an activation here. We will see how to model that in differential equations. But at the end, this is a graphical representation of a differential equation that is happening underneath. Okay, so I may have three different representations of this state of this model. So let's say I have the graphical representation. I know that, for instance, here, this U, that is a protein, activates or binds. This sign is prevent, so it inhibits this transcription factor, and then this transcription factor, because it is inhibited, will lower the level of Y. And how do I model that? Then what I can say is Y at t plus 1 is the negation of U at time t. So if time t, so these Boolean network models, so Boolean networks, the states are only 0 or 1. So if U is activated, Y will be repressed. And then I can go even finer. So I can go even finer because in reality, proteins are not on or off. So I have a low value, or I have a high value, and I have all possible concentrations in between. So actually what is happening is that there is a differential equation that controls the expression of Y, but this expression of Y, so Y decays over time. So this is this minus beta, it is degraded over time. And the proteasome, for instance, can degrade Y. And the expression of Y, so the increase of Y is related or is controlled by U. So U with some delay. That is why you have U t minus tau. So somehow the fact that this is t minus tau is the same as t minus 1 or t plus 1 t. So this is how you can control, you can delay the effect of U onto Y. So, and the higher the U, the smaller this velocity. So now you have three different kinds of representing the same information. You can do it in as a pictorially, so you make a picture of it. You may also, this is represented this way. And then you may do it in a kind of simplified word where the concentration of proteins can only take a couple of values, a couple of states. And then you have all the Boolean networks that we saw before, and these probabilistic changes that we saw. So these ones. So either you may think of this is U, this is Y, this is whatever, and then how they evolve over time. Or what is actually happening is that there is some chemical reaction, and this chemical reaction, I have to model it. Okay, so yeah, it looks something like this. So let's say that, I don't know from where I've taken this. I think it is the same paper as before. So in a simplified world, my transcription factor, so for instance, goes from low to high, low and high, and so on. And these kind of equations describe the dynamics. Okay, it is not a static, it depends on time, but the dependence on time is oversimplified. What is happening in reality is that I don't have just two states, I have a whole continuum of states. But this whole continuum, I can approximate by discrete states. Okay? But What is in reality happening is this change, these smooth changes controlled by the differential equations that are associated to the chemical reactions. Okay, and for instance, oversimplified networks still can be useful for many things, especially when you have many networks that is relatively complex with many different proteins or concentrations that you're looking at. It can be difficult to get all the parameters for those, and then you can you may come with simplified models. So, for instance, this very simple model, which is here it is called recursive deterministic model. Okay, so let's see what it does. So it is deterministic because if you know the state now, if you know the state at time n, you know how to calculate the state at time n+1, and that will be a function that somehow controls that. And this function can be as simple as this one. Okay, so you take the current state. So you have this node. This node is connected to a few other nodes, and you will have only two kinds of connections, either green or red. Green is +1, red is -1. So if green means if this protein is activated or this gene is activated, this one will be activated. Red is if this gene is activated, this one will be inhibited in the next time. And then you do the following. So you are at the current state, you take this node, you see all the connections, and you make this sum. So the states are either 0 or 1. So either they are 0 are inhibited or they are activated. And then these A's, they are also +1, -1. So this sum is either positive or negative, and it is extremely simple to calculate. And if it is 0, it stays as it is. So if it is negative, you will be inhibited. If it is positive, you will be activated. And then you let it evolve. Okay, so is you let it evolve. And for instance, here in this paper, what they found is that they look at a few proteins of interest. So some proteins that are related to the phase of the cell. So the cell has a few states. So it's that it has G1, that is growth, and then DNA synthesis, that is state S, and then in state G2, that is growth and preparation for mitosis, and then you have the mitosis. So by looking at these, I don't know, 10 proteins that are interested, and by defining what are the relationships between those, so these arrows, they are able to simulate, and they see that if they start at some particular state, then it goes to G1, and then it goes to S, and then it goes to G2, and then it goes to M, and then it goes again to G1. So this is a really powerful, even if the model is very simple. And also what they show is that, okay, so we have started in a particular, and then it stays, okay? So it stays in G1 for a long time. And we have started from this combination. This combination, if you have this is a node. This is one of these nodes. Okay? So it is a node. Let's say you have 10 of those. It's a node like this, but now you have 10 bits in your state. And now what they did was, okay, let us start from random nodes. If I start from a random node, where do I go? And they follow the path, and they go always to G1. So somehow these gene networks, they are designed by nature, not by these researchers. the researchers, what they did was to propose how they are connected. But by nature, they are designed in such a way that if you somehow get away from G1 state, you get away by for whatever reason, you come back to G1. So you remember homeostasis. This is like homeostasis somehow. You are in a state, you repeat there, and then you go back to the same state. Okay, and what if we can, this function here is not deterministic as in the previous case, but it is a stochastic. So for instance, here in this paper, what they do is, okay, I will try to model how populations grow over time. And then given the current state, so the current state is given by the current size of the population, I will compute the next one. But then I will have some other, this is a not open system, but yeah, if you consider births and deaths as going out from the system, then it is an open system. And then you have the migration also, if you consider this as a country. So now it is an open system. Okay, so now births, for instance, here, it will have a fertility rate. So there is at a particular country, C, and a given time, T, you will have a total fertility rate. Okay, so somehow how many children you have in a given year. And this will be the same as the year before, minus something that that is a decline that they have chosen to be deterministic somehow, plus some noise. Okay, so I cannot predict exactly what the fertility rate will be this year, and then I put some random noise in there. And now, if I let the system evolve, I will have a deterministic path. What I have is a random path. But this random path, you may remember from last lecture, where we had random collisions between two atoms of hydrogen to form a molecule. So the path was random, but the average of those paths was the deterministic. So deterministic models, like these ones, they are normally approximations to reality, what is happening that is stochastic. And the average of all those stochastic realizations, they give you more or less the trend that the system is following. And then, for instance, here in this paper, they made projections of what is the population, the size of the population in the world for different years. And this is also something interesting in general in models. So your models can be very accurate for the kind of data you have seen. So you look for the parameters. So this is here. This has the parameters. This theta of the country, they are numbers. And you estimate those numbers. And then within the region where you have data, they can be quite accurate. As you go away, there's more and more uncertainty. And it is important that you model the uncertainty. Okay, so this is also, have you heard of interpolation? And interpolation you have heard for sure. In image processing, we have done interpolation. And extrapolation. Have you heard of extrapolation? No? Okay, so let's say that you have a function. So you have a function like this. You have some points. And this is y, this is x. And you want to construct f and y. We'll put a model like, for instance, let's say a0 plus a1x plus a2x squared plus some noise. And then my model has to estimate these parameters. Once I have estimated the parameters, I will have a model that says, okay, this is your model. Within So within the region where you have data from here to there, no, this is X, to there. Okay, so within this region, I am interpolating. My model, if I ask the model, okay, what is the value here? I am interpolating and saying, you know, this is your value, your predicted value. This will be Y predicted. But outside this area, outside that area, from here and there, I am extrapolating. So extra is outside, inter is inside. Okay, so models are valid or are only valid within the range of data that you have used to find the parameters. Outside that, they can be rather both. Not always, okay, but they can be. And it is important that you associate somehow of uncertainty to how sure you are that your predictions are good. And these are calculated, these are not, okay, I put this now, you have to calculate that. Okay, different kind of models, these Markov models. So Markov models, they also are defined on states. And then with these states change over time, but they do it in a probabilistic way. So let's say that you are in the starting state, and then with probability 0.6, you go to a rainy day, and with probability 0.4, you go to a sunny day. Let's say you are in England. Okay, so if today is raining, what is the probability that tomorrow will be also raining? Let's say it is 0.7. And if it is not raining tomorrow, then it will be sunny. So if this is 0.7, this has to be 0.3. And then if today it is sunny, then tomorrow will be sunny again with probability 0.6. So you have to say which are the probabilities of the next state given the whole chain. So here you have a chain. So the first day, second day, third day, today, and then you have to say which is the state, the probability of each one of the states tomorrow. And a Markov, so this kind of sequences, they're random sequences, and it's a random process is Markov, so has the Markov property or Markovian property. If this probability, this is a conditional probability or conditional of tomorrow, given all the previous states. So if it only depends on today, so the state now contains all the information you need to calculate the probability of next state. Not all systems are Markovian, okay? So there are systems that fulfill this property, there are some other systems that do not. And then you will have a random sequence like this. Actually, this web page there, it has a very nice simulator. So click on here. But it explains a lot, but then play a bit. This is your model, and these are the prior probabilities. You see that the other one is adapted because the sum of the two probabilities has to be 1. Okay, so let's say we put it like this. And then if today is sunny, the probability of tomorrow being sunny is also quite high. If today is rainy, the probability of tomorrow being rainy, let's say this one. And then you can simulate. So you start and then you go sunny, 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 and rainy. Okay, so you are going randomly along these states. Okay, so nice model. Here there is some two important elements. The first one is the prior probability, that is this one. So that is the probability of being in any one of these states independent of time. So if you take very, very long sequences, which are the probabilities of each one of those two states. And what is the other one is the transition matrix. So the transition matrix is this one. So if today is we have sunny state, what is the probability of tomorrow having a sunny state and so on. So Markov models evolve the probability distributions. So the first one will be pi. Pi is not 3.14. Pi here is the probability distribution. So we will start with these probabilities. This will be pi naught, pi 0, and then we will calculate pi 1. And pi 1 will be pi 0 multiplied by 8. And now you have the probability distribution. From the probability distribution, you will get a random symbol. Okay? And so I know what is the probability of today of each one of the states. We'll choose randomly one of the two. So here, what is evolving over time is not the state, it's the probability distribution of the states, which is a change. It's a big change. And you can have hidden Markov models. So Markov models is this one. A hidden Markov model is this. So let's say that you cannot look up to the sky to see whether it is rainy or sunny. So you're inside your house and all you see is somebody coming in, and this somebody is either wet or dry. And so there are some hidden states that you cannot look at. And if it is a rainy day, the probability that that person is wet is 0.9. If today it is raining, the probability that that person enters dry is 0.1. And you have the corresponding probabilities for sun. So the state is here, but you cannot observe the state. What you can do is to observe some observables. So these observables, you also need to model how this process happens. So that will be the standard Markov evolution. So this is the probability, the evolution of the probability distribution. But then you will also have an emission probability. So the emission probability is given any one of the states. It's the probability that I get these two states. So this is the emission probability, not for this example. If you want for that example, you would have to, it would be dry and wet. Yeah. And here it would be rainy and sunny. Okay. So you have the states evolve on their own, and at a given state, you have a given probability of observing some variables, some states. So you have to infer from the observables, you have to infer what is the internal state of the system. Here you see an example where this kind of stuff has been used. So a hidden Markov model to identify mutation signatures in cancer. So signatures are, okay, so there are some internal states of some proteins being overexpressed or underexpressed, and then how to use hidden Markov models to identify which are the proteins that are involved in that disease. Okay, next. Yeah, this is the same paper, I think. No, this is a different one. Yeah, this is interesting. One of the paper, this paper identified this protein as being related to cancer. And this other paper, they show that if you mutate this gene, then it actually causes cancer. Okay, so one of the signatures found by the hidden Markov models, it was confirmed by some other studies. Okay, software to work with Markov models. Here we have a few. Yeah, something different with respect to the ones before. Now you have to program all this stuff. You have to really write programs to use them. Yeah, some other more software. Yeah. Okay, going back to differential equations. So I told you before, okay, so we will see how you can express that something is inhibited or it is promoted. Okay, so let's say that you have this kind of graphical representation where you have a few genes. So let's say you have these genes, and you have relationships between those genes that somehow, if there is the DNA damage, this RECA is activated, and RECA activated implies that LAC-R is deactivated or whatever. Okay, so you have to go from a pictorial representation to a more mathematical model. And in this mathematical model, you will define the concentrations of proteins of interest. So we will define these three that are the ones that we are interested, X1, X2, and X3. But there are also other two that are somehow involved. These two are the proteins. So we will also have X4 and X5. Okay, so what are the relationships? Okay, so let's look at X3. I think X3 is the simplest one. So X3 here. So X3, there is an input from the outside, that is whether there is a damage or not. So if there is DNA damage, then this value, let's say that this protein, whatever that is, or that metabolite or whatever, it goes up to a concentration of 25. If there is no damage, there is it has a value of 1. So this is a promoter. If this grows, X3 has to grow. Okay. So if this grows, X4 grows, X1 has to grow. So I will put a positive because it has a promotion effect. I will use a positive power. But this is somehow mediated by this protein. So it is not the damage alone. This is the damage with the help of lambda2 of this lambda CI. So this lambda CI will have to multiply this one. And this one is somehow, it has an inhibitory effect. So we'll use a minus power there. Okay, yeah. No, sorry, I was reading the wrong equation. It is this one. We are talking about X3. So X3, it has X6 to power, and then some coefficient, and then it degrades. Okay, that's fine. Okay, now this like X2. This X2, somehow it is affected by X5 in a positive way. It is affected by X1 in a negative way, and it has some coefficient. And then it degrades. And the degradation of this, goes. The degradation of X2 somehow is influenced by X3. Where is that? Yeah, here. X3 affects the degradation of X2. So X3 affects the degradation of X2. And then we have X1. X1 is okay, so it is affected by X2 and X4. So the expression of X1 is affected by X2 and X4. Affects the degradation of X2. And then we have X1. X1 is okay, so it is affected by x2 and x4. So the expression of x1 is affected by x2 and x4. x2 has a negative effect, so it is inhibiting x1, and x4 is promoting x1, and then it degrades. We have the degradation. So now we have the equation system. You have to give some initial conditions, and you let the system evolve. You let the system evolve, and you have it here. And I haven't copied the notebooks to this computer. This is the class computer, so I haven't copied them. But the important parts are here. So you have to define, for instance, x6 is the external input. Okay, so let's say that x6, that I will say the function of x6, will be a step function. A step function that goes from 60 to 60 to 600. So from 60 to 600, there will be some damage. Okay, so it goes from 0 to 25, from 1 to 25 in this range. Okay, then what happens with the rest? Here I'm just showing x1 and x2, just not to make it too cluttered. So you have to tell what is the derivative. We did this the last day. So in the last day, you have to write this equation system. This part, the right hand side is f. So you have to write it, and then it is in the notebook. You can give it to the solver, and then the solver solve this differential equation with these initial conditions that they are not shown here, but they are in the code. And then these are the responses. So these responses, let's see if they go as they should. So when there is damage, happens, x1 grows, and then x1 grows, that is this one. The damage is repaired, and at some point it goes, the damage disappears, and x2 is repressed during that time. And then when the damage disappears, x1 is deactivated and x2 is activated again. And this is the kind of work or any one of these papers that you are reading or whatever, that if you want this extra point, you have to do by yourself. Okay, I'm giving you a lot of notebooks, so you can use them as basis. Okay, and yeah, so why there is fractional power? Fractional powers are new in chemical reactions, in our models of chemical reactions. And these fractional reaction rates, there are a number of reasons for that. Yeah, at the end, not everything, that there is some kind of inefficiencies, and these inefficiencies that are there. I got all these texts from ChatGPT. Okay, I don't trust it just but I read it and it looked very. Most of the reasons I knew, they were there, and there were some other reasons I didn't. Okay, what kind of inputs can have? So we can have pulses, as we have had with the DNA damage. You can also have, this is my name, this is a ramp function, this is not mine, but infusion with ramp. So infusion, for instance, when you are administering some drug, you can have an infusion. And these infusions, they're a constant, but then you make and you have a rise. Okay? You may have an infusion. This is in a step function, or you can have something that is called a bolus. Bolus is just the delta. Okay? No, actually, you recognize the name systems biology. You have had a course, system signals and systems, right? And you have had this kind of inputs to them. So yeah, an example of a gene regulatory network. So let's say that X1 and X2, they are controlling each other and with this sign. So one of them is inhibiting, X2 is inhibiting X1, X1 is promoting X2. And yeah, X1 is promoting itself and X1 is promoting X2. So I can write all that here. So X1 is promoting itself, X2 is inhibiting X1, and then X1 is decaying. So it is degrading. And then we have the decay of X2, and that X1 is promoting X2. So somehow that reflects those relationships. And let's analyze the behavior of this system. So it is a kind of feedback system. X1 promotes X2 and itself, and X2 inhibits X1. So now I will choose a couple of constants there, alpha 0.9, 1.02. So you can see that this one goes to damped oscillation. And okay, so it goes to some kind of a steady state. If alpha is 1.02, it doesn't go to a steady state. It goes to an oscillatory behavior. So you were asking before about circadian rhythm. So this is an structure that leads to cycles. And yeah, so here, for instance, we have this equation system. This has nothing to do with genes and proteins. So let's say that we have 3 variables, and this is the equation system. And for particular combinations of the constants here are rho, sigma, and beta. So for particular combinations of them, what we see are different trajectories. So you start at a point, and then it goes. And what you see, for instance, here you have also, you start at this point, and what is the trajectory? You start at this point, what is the trajectory? You start at this point, what is the trajectory? And you see that at the end, you have kind of stable cycles. So this stable cycling is again a little bit like this homeostasis, but not homeostasis, but kind of stable oscillations. So it doesn't matter if you perturb the system, it goes to some oscillations that are regular. And yeah, does it happen? So these kind of states, that somehow they are particular. So in this phase plane, you have that they oscillate around points, or they may go to points. Sometimes they also go to points. Those points that attract all the others. So it doesn't matter where you start. At the end, you go to that point, or a cycle around that point. They're called attractors. So these attractors, they also happen in biochemical networks. So when you have for instance, this is you have a gene network, and you have 2 different states. So one that is the healthy state, and the other one that is the cancer state. So, and you may have different cancer states. Actually, cancer is not just a disease; it is a whole collection of diseases. Okay, so there are many different kinds of tumors. So you may have many different attractors, and what you have to find is a way of perturbing the system so as to make the concentrations to go to the healthy state. And here we have a paper describing these attractors in cancer networks. And yeah, any question up to here? Okay, so let's see the next lecture in the book. There is this parameter estimation. the parameters, we have seen that in all these models, you have parameters, and depending on the parameter that you choose, you will have a different response of the system. And you may have different problems there. One is simulated, so given the parameters, tell me the output. The other one is the opposite. Given the output, tell me the parameter. So I have observed this. It is a problem more similar to this one. I have observed this point. Tell me the parameters that are consistent with those points. And for that, you will use optimization. So we will concentrate now in the parameter estimation problem. So the parameter estimation is, for instance, let's say that I am observing how bacteria culture is growing over time. So somehow I measure the size, the population size with respect to the beginning, and I will put it in logarithmic units. And I have two different input variables. One is time, so how it grows over time, and how it depends on temperature. So I have here I don't have the different possibilities, but you have for different times, you would have for the same time, you would have different temperatures also. Okay, with respect to some set level. So you have this is the delta temperature. Okay, so now what is my model? So I can say, okay, my model is this output here, this output here, this is yi, will be a function of this, will be a function of time. And this function of time will depend also on the temperature. So at the end, I will have a couple of parameters. So I will have beta naught, beta 1, and my model is this. Okay, so, and these are my input. My input are time and delta T and delta temperature. So which are the parameters? So what I have to do is to play with beta naught and beta 1 and try to make this difference as small as possible. Okay, so this is my prediction. This is what I observed. And I have to make this difference as small as possible. So I have to find beta naught and beta 1 that makes this smaller. So that is the optimization problem I have to solve. And here we have used a quadratic difference. Can I use anything else? Yeah, there are many others. So you can use this mean square error, that is the one before. Mean square error is the difference between what you observe and what you predict, the square. And some other possibility is this absolute value. Both have different assumptions, statistical assumptions that we will go into, but they're very interesting. If you are interested, look for this kind of stuff on Bayesian statistics. But there is a problem. The problem is this function here. As you go away from the observed value, it grows very quickly. This one grows much more slowly. Here you have the different curves on the same scale. So this one is growing very quickly as you go away. This one is growing more slowly. And for instance, this Haber distance is part of it is quadratic, and then from some point on it is linear. And it has advantages because if you keep the quadratic one, what happens is if you have an outlier, this outlier drives very much the estimation of the parameter. Okay, and here you have this example that can you see it with the light? Yes. Okay, so a possibility. So I have to look for beta naught and beta 1. So something I can do is I know that beta naught and beta 1 are somewhere here in this range from 0 to 0.005. So I explore possible combinations. I compute the goal function. I compute this thing. I compute this one, and I keep the one that is minimum. Okay. I plot it, I keep the one that is minimum, and it goes down to, I think it is this one, 0.002, the true value is this one, 0.002, 0.001. But 0.001 is outside of, yeah, it goes to 0.005. Okay, so this is the step. So it is away from, I don't have enough resolution. My steps here, they're too wide to see the 0.001. Okay, so exhaustive search is expensive. And also it depends on your step size. If your step size is not big, small enough, you will not find the true parameter. Okay, let's make a break if you want. We have seen that the exhaustive search is a possibility to look for beta 0, beta 1, but it has two problems. First, that I have to explore many different combinations of beta 0 and beta 1. The second one is that it is limited by the sampling. If my sampling is too large with respect to the precision I want, then I may miss the true parameters. So there is an alternative that is this gradient descent. Gradient descent is a continuous algorithm, so it is not limited by the sampling anymore, and it is iterative. So you start at the current guess of the solution. So you say, I don't know, beta 0 will be 0.03 and beta 1 0.03. So you are somewhere here. And then from there, you go where this surface goes down. But there is a kind of catch. This surface, but I don't know the surface. All I know is the function at a point. How do I know the surface? Because the surface I know because I have done sort of search, but otherwise I don't know the surface. What I know is the function. I know how to evaluate that function. What you need is to know how to evaluate the gradient of that function. So you don't need the surface, but what you need is the gradient of that surface. So in a way, the gradient, what you're saying is the following. So you have a point, so let's say that you have here beta 0, beta 1. For every one of them, you have a mean square error. Say that it has some surface, I don't know which one it is. Okay, but it has some surface there. And all you have is a point. And that point there, you know what is, so you have an estimate that is your current estimate, let's call it 0. My current estimate 0. That is this one here. And I know what the MSE of beta 0 is. That I know. Okay, but I know I need to know how to compute derivatives. So the derivatives somehow is approximating the surface locally by a plane. And then it looks where the plane is going down, is going to. And then it will move, okay, the plane is going in that direction. So my next move, let's move red. So my next move will be in this direction. That is the direction that the plane is telling me. Okay, so yeah, that plane, if you have to define a plane, what do you need? You need the normal of the plane. You need a point that the plane goes through. That point I know, this one. And you need the normal to the plane. So you need this vector. And that is the gradient. The gradient is that is somehow telling you what is the plane. So you are approximating this by plane, and then I will go from where I am plus a little bit. And this little bit is determined by the gradient of the error function and multiplied by a step, by a small step. Okay, what is the gradient? Okay, so this is the error function. I can write it this way. So this is the difference, a square difference between my observations and my predictions. My predictions look like this. And this E is a function of the observed data. This is observed, and this is observed too. The green one is observed. And my parameters are beta 0 and beta 1. So actually, the observed data I cannot touch. It is what it is. But I can touch the beta 0 and beta 1 and make this E to be as small as possible. So this is a function of beta 0, beta 1. So I can compute the derivative of E with respect to beta 0. So derivative, this is a sum of things. So it will be the sum of the derivatives. So it will be the derivative of E with respect to this particular error. So I have this table here. For every entry in the table, I'm committing an error. So this is this error here. So this is the error that you commit for each one of the entries, and the derivative of that error with respect to beta 0. Okay, if this is the error, this is the error. Okay, so this is my observation, this is my prediction, this is the error. Derivative of E with respect to the error. It is twice I. And then derivative of the error with respect to beta 0 minus I. There it goes. And here I know everything. This is my error that I know what it is because I have a current guess. And it depends on something that I have measured. Okay, what about beta 1? Beta 1 is the same, but now the derivative is different. So this is the derivative with respect to beta 1. Have it there. And this is the gradient. So now all these things here are just two numbers. And then I can add to my current guess of the two numbers, of the two parameters. And to make the thing stable, I have to this because the plane is not the surface, so it is only an approximation. I cannot go too far because if I go too far, probably the plane is deviating from the surface. So I just go a little step in the direction that the gradient is telling me. The gradient is the direction of a steepest descent. Okay, so if you are at the point, you look all the directions around. So you have to do it this way. I have this point. I look for all points around. So I look for all points around here. I evaluate here. For each one of them, I have a point in the surface. The one that gives me the largest change is the one of the gradient. So the one of the gradient, what's this color, is the one that tells me in which direction I should move. It is the one that goes steepest in the surface. So yeah, so now it is iterative. I'm not limited by the sampling step anymore. And there you have it. So mu, this is this constant there. Number of iterations, this is your initial step. And then you have to do what you have said you will do. So you make a prediction, you compute the difference, and then you compute what is this is a vector here. So you compute what is the sum of this difference squared. And then you start with the gradient to 0. Actually, this is a mistake, isn't it? This is a mistake. Yeah, this is a mistake. It should be 0. But you see, this pretty goes to small mistakes. So you predict, you compute the errors. Now you accumulate all those errors according to the formula we had before, and then you move the current guess. You move it a bit according to the gradient. You let it iterate forever, not forever, for 100 iterations, and then at the end you go to here. And it's pretty close to the ground truth. So what we have seen is, okay, depending on the parameters I choose, my models will give me different responses. So given the observations, I need to find a way of finding those parameters. And one possible way is an exhaustive search. And now we have seen another one that is this gradient descent. Gradient descent has a drawback too, so it is not a perfect tool. So it has a drawback that if you have local minima, gets stuck in those local minima. I'm not showing that here, but we can do it here. Sorry, we can. So let's say I will do it in one millimeter because it is simpler. So let's say that we have a single parameter, because here we have the MSE. Let's say that MSE looks like this. So depending on your beta zero, you have different values. And if you start here, you are fine. You are fine. You will go down to the global minimum. But if you start here, you are not fine. You will go to there. That is the steepest descent direction. So okay, so then for some particular problems, it is rather easy. And this is one of them. So there are problems for which there exists an analytical solution. So in general, there is not. But for some problems, and linear problems are one of those, you have an analytical solution. So you have this model. You have this model, and it has to be linear in beta. Okay, so it is not linear in t, for instance. Well, in t, it is also linear. Okay, so this model is linear in everything. But for instance, this one that we saw before, this model here, it is too much. It is linear in beta, in h, and my parameters are called A. So it is linear in the AIs, but it is non-linear in x. Here's x squared. Okay, so a function can be linear in some parameters and it can be non-linear in some others. Okay, so this one is linear in everything. So in particular, in beta 0, beta 1, that are the ones we are interested. So that means that I can factor it out in a matrix form. So I have all this data that we had at the beginning. So these are our observations. And then, so we have our observations and our predictors. So you see P, so I have T1, T2, and I have T delta T. So I have T delta T for each one of the entries. So this is a matrix. So I can take this matrix and look for the least square solution. This already, you know how to get it. We did it last year in image processing. And so there is an analytical solution. You do the analytical solution and you get the right answer. But not always you have analytical solutions for your parameters. Very often you have all these kind of landscapes. So you would like these smooth, convex landscapes where it doesn't matter where you start, you will go to the global minimum. But reality is more complicated, and when you use this, when you lose convexity, what happens is that there are kind of valleys around, and then it depends. This is called non-convex. And then what happens is that you may get trapped into local minima. So it depends where you start. You may go to different local minima. Okay, so why don't we look for algorithms that are better at getting away from local minima? So for instance, here, let's say that this is my cost function. I want to go there, but gradient descent would go in this direction, okay? And it would go there. But why don't, for instance, if I'm here, I would never find the global minimum. Okay, so why don't we allow for big jumps? So we will do the following. So I will start at a point. I will take a point in the neighborhood, and I have to define the neighborhood somehow. But I pick a point in the neighborhood, and this neighborhood can be as wide as this one. And I evaluate the function there. If the function is better than mine, I keep that point. Okay, so if the function is better than mine, then you keep it. Okay? It's not there. But so you're here. This is your value. Now I pick a random point in my neighborhood. This one. Is it better than what I had? Yes. You keep it. You stay. Okay. Good. Now you take another point in the neighborhood. Let's say that you take this one. Is the function better than before? No. Gradient descent wouldn't take it. Gradient descent says, okay, this is worse, so I stay where I am. But this algorithm here is called simulated annealing. And simulated annealing, that's something strange. So simulated annealing says, okay, you may still go to something that is worse, but with a given probability. And this probability will depend on a temperature. So if you are too far, so let's say that we do it in this way. So now I'm worse, okay? So I am, let's do it here. Okay, so we have a couple of points. We have this point, and this is the new suggestion. The new suggestion now is green. Sorry, it is not there. The new suggestion is here. It is worse. Okay, so there is a delta error. Okay, so I will do it. I will take it with something with a probability that is proportional to that. Let's see how it goes. Let's say that my temperature is very high. If my temperature is very high, this is very small. This ratio is very small. Okay, it is goes to 0. The exponential of 0 is 1, so I will take it. If my temperature is very high, I will take it. I don't care if it is worse. And that allows me, so instead of going into the right direction, now I'm going into the wrong direction. I'm going in the wrong direction. I'm going there. And here I do the same. I will take a point in the neighborhood. If it is better, I keep it. If it is not better, I take it with this probability. And the trick is that this probability depends over time. And with time, this probability goes down. So at the end, I will not take back suggestions. So at the end, with time, you see the temperature goes down. With K is the iteration. And here there is a video that shows how. Doesn't show. Okay. So there is a video. If you look for simulated annealing in YouTube, you will see this point starts, I think it starts somewhere there. It goes all the way around the surface, but at some point the temperature starts to be very low, and then it goes to the global minimum. It only takes good suggestions. It is called simulated annealing because it tries to simulate how metals cool. When you want to bend a metal, you first heat it, and then you can bend it, and then it cools. And in the cooling process, the energy of the metal is minimized, and they simulate that process. Something else, genetic algorithms. Yeah, it is I will connect my computer later, maybe, and I'll show you these videos. So genetic algorithms are kind of similar. Similar in the sense that they are stochastic. So they will start with a population of individuals. So I will have a population of individuals. And this is my population, okay? I have 4 individuals. And these are my parameters. Here in this example, the parameters are just binary, 0 or 1, right? They can be real numbers if you want. And then each one of the component of my individuals will be called a gene. And the whole thing will be a chromosome. So I have a population. This population, what it's doing is sampling this. So let me do it here. So it is sampling the surface. So we will have theta 0, theta 1, and the 0. We have some surface here. And what we have is a number of suggestions, random. So each one of them sees how good they are. Okay, so now we will do the following. We will take randomly, so the best you keep, so the best elements here. So let's say that you have a population of 100, so you keep the first 5. And for the others, they will create a new generation of the population. And for that, you will take 2 of them at random. You will split the chromosome in 2 parts at random again, and then they will exchange. So for instance, these two, this is the father, this is the mother. I split there, and then I create 2 children that will be these 2 that are flipped. So when you are close to the minimum, this flipping and these exchanges, they don't care. So if they start converging to the right place, I don't care if you mix them. They will be more or less all of them in the same place. And also you can simulate mutations. So you can take one of the genes at random and change it in a crazy way. So you change it, and now what you are doing is something similar to simulating an alien. You are taking it away, but maybe taking it away brings you to the right minimum. So far for the optimization. So now we have optimized. We get a value. How sure you are that that value is the ground truth? So you have some uncertainty on it, and this uncertainty depends also on. It depends on the amount of noise you have. If you have a lot of noise, so your observations are very noisy, then you will have more uncertainty of what your parameters are. And if you have less noise, then you will be more sure. So at the end, what you will have is for each one of the parameters, it doesn't matter what they are, is Virginica or reciprocal or whatever. So for each one of the parameters, you will have an uncertainty, and you will have a distribution. So this distribution is, okay, I have observed this red one, but it could be any one of these within this range. And you construct a confidence interval around that parameter. And this confidence interval, you can do it if you assume that the residuals are Gaussian. You can do it in a parametric way. Have you constructed confidence intervals in statistics? Or yeah. So this is the same confidence interval that you did. And or otherwise, if your data, if your betas are not Gaussian, then you use bootstrapping. And the fact that you have uncertainty in your parameters translates that you have uncertainty in your predictions. So some predictions will be more accurate than some others. Here, I have more uncertainty. So my predictions would be more uncertain. And here you see that why I have more uncertainty. This is much more noisy than this one. Okay, good. Then optimization algorithms. We have seen a few of them. I think we have introduced most of the important ideas, but there are really, really a lot of algorithms to optimize. Here you have the names. And yeah, so here you have also another notebook. I cannot install Jupyter here, I think. And then, and if I put it in my computer, then you cannot, it is not recording when I use the whiteboard. Okay, so let's say that we have some observations. These ones actually are when you take a pill, this pill dissolves and it releases the drug that you are taking. So how much amount, when you take a pill of 1 milligram, how much amount is left changes over time. And it changes over time. Either you can describe it with a differential equation, how it changes over time, or for instance, you can solve the equation. The equation is solved like this, and then now you have a couple of parameters. So you have to look for K and the initial condition. The initial condition here in this problem, you know, it is 1 milligram. That is what you do. And then how I can fit that. So here we have a notebook to do that. So for instance, here, this one is using differential evolution. Differential evolution is one of these genetic algorithms. So I have to optimize the MSE. I have to give some region where the population can live. So they can live between 0 and 1. And then you can give parameters for the F. These are interesting thing for the F. So the MSE is first I simulate. Okay, so I simulate, and here is the derivative. Okay, so this F is this one. It's this one here. So you solve the differential equation with this initial value. Now this is the solution. Now I interpolate this solution at specific times with this step. And now I compute the difference between what I observed and the predicted one, compute the difference, and this is my error. And I pass this error to differential evolution. So all I have to give to the optimizer is how to compute the error and where the parameters live. And it will find a way. And here you have the evolution. So it starts with something completely crazy, very large error. And then every time I evaluate, I'm printing. So at the end, it goes down to very low error. And this is the 0.24 that is around the loop, or close to it. And now you can plot. I knew this is simulated data, should be 0.25. So you can see that there is an uncertainty. It is not exactly the right value, but it is pretty close. Any question? Okay, so next. So there are a number. So once you have the system, you have defined your system. So the system is what are the equations and what are the parameters. So once I know them, I can do different things, or I can solve different problems related to the system. So the first one is system analysis. So system analysis is use it to predict what will happen if I do this. The other one is system identification. System identification is the opposite. So it is given the data, which are the parameters and which is the structure of the system. And something else is control. So I want, for instance, this is used in diabetes. So people with diabetes, that you can monitor how the glucose is going over time. I have a model of how the body will react if I inject some insulin. And then, because I know how to predict, and I know what will happen, I can inject more or less to make the glucose to go to the level I want. That is control. But for control, I need a system that simulates. And then system design. System design is I'm not designing, I'm not analyzing a system in nature, but I'm designing a system. So, for instance, this synthetic biology. Have you heard of that? So, synthetic biology is I will create a bacteria that does this, and normally no bacteria that does that. But my genetically engineered bacteria will have some circuit, some genetic circuits, and some proteins that are we have seen them. So if it finds glucose, it will inhibit this protein, and then this protein will promote this other factor, and then it will whatever. So I inject that circuitry into the bacteria. Mathematics. So the whole point of systems biology is the mathematics behind it. So that is why you can talk about the biology that you like, but you have also to tell what is the mathematics behind those biology. Because the word systems is a modeling of how the system will behave. Questions? For instance, for your practice, you can go to any one of the students and see for an interesting topic that you like, and you can make that your practice. Any question?